Welcome to another video tutorial. In this video, I will use this Scispace tool to help you in writing your review of literature, whether you want to publish your review or whether you want to write a review of literature for your thesis. So let's try this AI tool and explore its potential, whether it can be helpful. So here is its website. You can see typeset.io. And let's discover any of your topic. Let's suppose my topic, and if I want to write a review on like what is the functional role of tapetum organelles, what is the functional rules of tapetum organelles in pollen development. Yeah, the good thing about this, you can see when you will be typing your questions or area of research, you can see it is also giving you suggestion whether what is the specific molecular mechanism by which the tapetum organelles regulate pollen wall development formation. So that's what exactly what I want to search. And here you can see it has already given you another option, which is, I think, very smart option of this AI tool. And you can see how do tapetum organelle influence the expression of gene involved in the pollen development, and similar, what is the role of tapetum organelle in the regulation of pollen grain morphology and its implication in pollen plant reproduction. So let's first try this question and let's try to get what actually it can tell us about that. So you see, actually now it is populating 5 answers, and you can see here are the answer from only 5 top papers. And let's try to read this, because this is area of my research, whether it is good or not. So you can see that tapetum plays a crucial role in pollen wall formation through a complex interplay of molecular mechanisms. The process involves the secretion of essential component and the regulation of developmental signal. Yeah, pretty good. And the tapetum functions and the pollen ball composition. So you can see tapetum cell secrete nutrients, proteins, lipids necessary for pollen wall development, particularly during the late stages of anther development. So here is our recent review, and yeah, that's actually, yeah, that's the right citation. And I know this paper, this is paper is published in Cell, and you can see this is comprehensive insights into the tapetum mediated pollen development in Arabidopsis thaliana. So that's quite good. And if we can see this second paper, actually this is OSLDDT1 encoding a transmembrane structural DUF 726 family protein, and is essential for tapetum degradation. It is not exactly like the tapetum organelles, but it's very relevant on the tapetum degradation. So I think so these first two examples are quite nice. And then we have molecular signaling pathways like receptor kinase and sulfated protein families, and facilitate the coordination of pollen ball development. Yeah, so now it has switched the attention from molecular mechanism of tapetum organelles to rather the molecular role of pollen wall formation and its regulated gene. So it is also a bit relevant. So we can try to read more, and here you can see we have actually the summary from 5 organelles, and I believe the first one is top most relevant, and other because this area is also like not very much explored. So you can also try other options of your own research. And let's now try to see if we want to add and want to see more paper. So as you see, here are the different options, and here are the some relevant papers, which this one is in Cell, and this is, yeah, this is also relevant. I mean, a peptide mediated multi-layer molecular dialogue for the coordination of pollen wall formation. And then we have PNAS, yeah, right, good. And Yeah, here you can see it has populated a list of relevant articles, so it can be very helpful while you are very new to the field and you don't know anything previously about that topic. So I think so this tool is going to be very helpful. And even we can add more columns in this. You can see if we only want to specifically look into the what is actually uh, that insight which has the author given related to this topic, and even we can add more columns like too long, do not read, and we can add, yeah, conclusion. Yeah, we are interested in the, yeah, we can go, even we can create our own columns, and we can add result section. We can also add research gap, and yeah, we can also add, yeah, future research and applications. Yeah, and from here we can maximum add 5 columns in the free version. So if we can go back to the pricing option, and as here you see, actually if you want to go with the basic version, you can see here you can have limited chat and limited literature review search, but it is still good to use. I mean, it would be helpful, and you can see it can add 5 columns. So if you don't want to buy, it's okay. But if you want to have a more control than the standard answer, so then of course you can buy a premium version per month. You can see, and you, if you belong to any university and you want your, if your lab will pay, so even this is the, like, the, for labs and universities it is even more cheaper. And if you are new and want to use this Scispace while watching my video, so here are actually few coupon codes you can apply to them, and it will give you like 20% discount for monthly plan and 10% discount for annual plan. And if you are from India, you can use this, and if you are from other countries, like, you can have 20% off or 40% off on annual plan, and you can use these coupon codes to get relevant discount. And let's go back. Actually, we have asked that question. Actually, it has populated a list of those papers which were relevant to this, more insights and conclusion results. And you can see the result shows tapetum regulate pollen development through the nutrient secretion and program cell death. Yeah, right. And here is actually the research gap of that study, and sometimes it takes a bit to populate few columns, so no problem, you can go and do something else while you will come back. I'm sure it would have generated something interesting for you. Explore few more feature, and as here you can see, if you want to add something, you can even develop your own notebook, I mean whatever you want, and even you can chat with your PDF. So let's try to explore this option, and as here you can see, we can upload any of our specific research article. I mean, if you don't want to go into the internet and you want to develop your personal library, so you can go there here and you can upload a PDF of this. Like in this example, I can upload my own paper, and as you see, we can just to name it as a new collection related to maybe anther development. Yeah, you can develop your own library as well. And now we can ask simple question using this tool, and as this Scispace simple question answers tool, and this tool is called actually the Copilot tool. So as you know, if you want to ask the standard questions, you can use this, but if you want to have a high quality response, so then of course you would need to buy the pro version. But let's simply using the standard version, so we can ask a few questions like, what is the main finding of this research article? Yeah, we can ask this, and try to know whether it is right or not. So as you can see, this paper is of mine, so I know what is actually the main finding. So actually the research article presents several key findings regarding the role of putative subtilation like serine protease. Yeah, actually I have cloned this gene. So as I know and I can judge from the headings that it is involved in the anthocutical biosynthesis, and it has impact on the apical spikelets, and it has gene expression changes. Yeah, and then we have conservation of function because the subtilation function is conserved in other species. Then I have genetic analysis. Yeah, actually this gene was cloned from a mutant, and that mutant has actually SNP. So I think so it has generated a very nice summary even using the standard version. So if we want to go and prepare, you can write what is the material used in this study. Yeah, so you see here it has given the rice cultivar name. You see the rice cultivar name was actually the Ishiang B and a Japonica, and then I have used F2 population, and then even it has given the information on experimental location. So I think so it is very nice. I mean, if you want to write your review of literature and you don't have enough time to go into the full detail, and really this Scispace tool and this Copilot tool especially will help you to extract the relevant information. And here is one more feature. If you want to add, like if you want to have a specific detail on anything, like we can explore something. Okay, like here if you want to explain what is program cell death, so you can select this one, and you can see here are the actually the different options like explain text, and you can even get the related papers and you can highlight. So if you want to get the related paper to this, so let me explain this text. Okay, what is actually this? So you can see explain the text program cell death, so it will try to generate a response against this query. So you can see that actually the program cell death is a natural process happening in living organism, and it is important for growth and development and living in our body. Yeah, but actually it is relevant to plant, but it's okay because the process is like journal, so it is giving the example from the internet and even quoting some examples from humans and diseases. So maybe we can ask a specific question like explain the text PCD in plants. Yeah, maybe now it will generate few more and exact answer. So you can see the program cell death is actually the biological process and involve the controlled death of the cells. I think so this tool is really helpful, and it would be very handy for those who are especially switching to molecular biology, and they don't have enough time to write the literature review of their thesis when you are running short of time.